I'm Susan Grew, and I want to welcome you. I'm the Director of Programs for the Newport Beach Public Library Foundation, and Medicine in Our Backyard is our series for the Newport Beach Public Library. Our foundation prides itself on supporting this wonderful library in its mission to be the cultural, educational, and informational heart of the city of Newport Beach. This lecture series definitely fits that billing, but we cannot do that without your help. We are completely membership driven and we receive no city funding. Please help us to continue to support this library by becoming a member of the foundation tonight. If you are not already a member, I see so many familiar faces here, and I thank you all for your membership support. And I encourage those of you who are not yet a part of our foundation to join. In the back, we have our newsletter, which lists all of our wonderful programs that are upcoming. If you are a membership, one of the perks of membership is you get discounted tickets for all of our programs. So you can find them listed here along with a membership envelope. And you can also quickly and easily join the foundation through our memberships website, nbplfoundation.org. And the web address is also in our newsletter. Tonight, I would like to extend a warm thank you to the Kempler family, Dr. Ike and Ginny Kempler specifically, for their sponsorship of this series. And I would also like to thank our friends at UC Irvine Health for bringing their extraordinary group of doctors here to the Newport Beach Public Library. This series, Medicine in Our Backyard, will continue through May, and the next lecture will be held on February 26th with Dr. Arvind Janab. He is the Director of Naturopathic Medicine at UCI Health, and he will be imparting some secrets to healthy aging, so you don't want to miss our February lecture. But before Dr. Janab, I have the distinct pleasure of introducing Dr. Leslie Thompson. Dr. Thompson is a professor of psychiatry and human behavior and biological chemistry in the UCI School of Medicine. She is also a professor of neurobiology and behavior in the UCI Ayala School of Biological Sciences. She is also affiliated with UCI Mind and the Sue and Bill Gross Stem Cell Research Program. These are two powerhouses in the field of medical research. In my own research of Dr. Thompson for tonight, however, I found another affiliation of hers that I think speaks volumes about her, not only as a scientist and researcher, but also as a human being. Dr. Thompson was profiled by student Jonathan Cohen for the My Hero Project. The My Hero Project uses media, art, and technology to celebrate the best of humanity, told in the form of a story. Jonathan writes his story about his hero, Dr. Thompson. According to Jonathan, Dr. Thompson, quote, wanted to make a difference by working on genetic diseases that affected people. She wanted to put all of her knowledge to good use. According to Jonathan, Dr. Thompson has brought the insights she gained in the laboratory out into the larger world, into the community of her patients and their families. Because for many of her patients, hope is in short supply. Jonathan says that Dr. Thompson is uniquely qualified to tell these patients and their families that there is indeed hope, that there is a community of researchers around the world trying as hard as they can to help them. Dr. Thompson sits with them, she talks to them, and sometimes she hugs them. According to Dr. Thompson, they are her family. I have to take care of them. I have to help them. We're eager to hear about your findings now, Dr. Thompson, but we are grateful to Jonathan for giving us a glimpse into your heart first. Thank you.
Thank you so much for that really kind introduction and for the invitation to be here. It's really a pleasure. No, I'm not Howard Federoff. But what I'm going to talk about is the same thing he was going to be speaking about with precision medicine and neurodegenerative diseases. And try to sort of give you a glimpse of some of the ways we're trying to approach these diseases as a whole and come up with better ways to treat them, because they all are very devastating and they are impacting larger and larger pieces of our population. So first of all, what is neurodegeneration? And this is the progressive loss of structure, function, or function of neurons, including the death of neurons. So it affects neurons in the brain and leads to the slow and progressive change in those neurons and dysfunction of those. And if you think about, so normally a healthy neuron will have connections to each other. They'll communicate with each other and they keep the brain nice and healthy. They have what are called neurotransmitters, signals that connect each other and tell the body what to do, elicit thought, elicit feelings, all of those. And they're connected with each other, and they have these electrical signals that transmit along what are called axons. In neurodegenerative disease, what starts to happen is these connections, these communication, they start to break down. As the cell gets sicker and sicker, they no longer can talk to each other and do what they're supposed to be doing, until ultimately some parts of the brain will, those neurons will die. So what we'd like to do is prevent that in some way. And this is just an illustration. I'll be talking a lot about Huntington's disease today. This is an autopsy material, a brain from somebody who died of other causes, was unaffected with Huntington's, and someone who had died from Huntington's disease. And you can see this big enlargement here. This is just space. There's nothing there. Just fluid goes through there, the ventricles. And loss of general structure of the brain, and that's what happens over time progressively. So this has become a huge challenge, though, to try to understand. It's very, very prevalent in our population. The major risk factor for this is age. So all of these are age-related diseases: Alzheimer's disease, Parkinson's, ALS, Huntington's. But we don't have a single disease-modifying treatment for any of them. We have some things that will help symptoms, that will alleviate certain things, but nothing that really slows the progression of the disease right now. And this really comes down to when you go into a clinical trial, you have a general lack of efficacy. So the treatments just do not work. So you might have seen them work in an animal model or in a cell culture model, but once they got into the patient population, they didn't work. And this shown here, this is about 56% of the cause of failures of drug trials is because of this lack of efficacy. Things like safety, strategies, commercialization, things like that also will impede these trials. But it really suggests that maybe we don't have the right target yet. We don't know quite what to treat yet for those diseases. And so, for instance, for something like Alzheimer's disease or Parkinson's disease or Lewy body dementia, is this one disease or many? And so you have a large population that has this disorder. But what if that what you have really are subsets of individuals and they share a cell function problem? So out of that whole, there might be a handful that have one type of problem in their cells, but they have symptoms of Alzheimer's. Another group has a different type of symptoms in their cells, but they also look like they have Alzheimer's disease. But if you're treating that whole group, you might not get a signal then, because you're treating everyone the same way with the same drug. So all patients with the same diagnosis are not necessarily the same. So subgroups need different approaches to their management and their therapy. So what we really want to try to do is be able to separate these out, to understand what these different subtypes might be for Alzheimer's or Parkinson's or ALS. And then that might help us treat these diseases more effectively. So what is precision medicine? This is sort of a catchphrase that's been, I'm sure everybody here has heard this term, right, that is in the press quite often. But this really is customization of healthcare with medical decisions, treatments, practices, products. Everything is tailored to the individual. So it's precise. Is the definition of this. And there was a precision medicine initiative in 2015 by the US government to enable a new era of medicine through research, technology, and policies that empower patients, researchers, providers to work together towards development of individualized care. And this was highlighted. And this initiative was funded then in 2015, and actually UCI is a site for one of this called All of Us, to try to get as much information as we can about the genetics of individuals. So data, lots and lots and lots of data, is going to be the foundation for this. And so we can use things like electronic medical records from over years and years and years of those medical records existing, say at UCI. There's been thousands of individuals with Alzheimer's who have been seen there. Can we look at those medical records and try to identify trends or patterns or things that might predict the course of the disease? Can we understand what's going on at a molecular and genetic level? So it can integrate the basic sciences with the clinical discovery and try to identify new treatments for clinical medicine from understanding at the level of the biomedical research. And one of the tools that we have at UCI is this wonderful UCI Genomics High-Throughput facility. The director, Suzanne Sandmeyer, who's here in the audience. And this has really facilitated our ability to get big data, to use genomics and DNA sequencing and RNA sequencing and really define everything that's going on inside the cell to get a signature of that particular cell. And so that's really facilitated the ability to do this kind of research. And as I'm sure all of you know, in September 18, 2017, there was a large gift from the Samueli family, from Susan and Henry Samueli, of $200 million to try to really use this type of integrative health, to understand what keeps people well and what can we do to prevent people from getting sick, and what do we do if somebody does get sick, and to integrate all of that information into what's called integrative health. And what we're doing now is we're just getting ready to launch a new program. It's a new center for precision and population health. And both myself and Suzanne are involved in this. And this is to then use this paradigm, use this structure to be able to get information from the electronic medical records through UCI Health, working with the physicians directly, feeding that information back and forth, having other investigators on campus be able to use that information, so somebody studying Parkinson's disease can then be connected with the patient records and understand more about what's going on and to take their mechanisms they're studying, what they're doing in the lab, and apply it to the patients, and vice versa. Learn from that patient information and apply it to the research. Generating biorepositories and tissue banks so that you have blood samples or tissues of when somebody has passed away from them that you can then incorporate into this whole model. Generate cell lines from patients. I'll get into this, the stem cell models that you can use to then do investigative research and identify what's going on in those particular cells. And this is a real collaborative effort with many other centers on campus, the Stem Cell Research Center, UCI Mind, which I'll talk a little bit about. The computing aspects of all of this, there's tons and tons of data that's involved. And somewhere you have to store this and you have to be able to work with this. So we are working very closely with people who are computational biologists or computational scientists. The Institute for Clinical and Translational Science at UCI is also a significant resource for this type of approach. And then once you understand some of the things on the molecular basis, you want to find out if they're true. Go back to those people, their tissues, to their medical records. Does this play out? And then ultimately identify individualized detection, prevention, and cures. And this really is the brainchild of Dr. Fedorov. This is something that he came to UCI with a vision to really put together an effort for precision medicine. So I'm going to give you a couple of examples of how we're doing this in my own research and as part of large national efforts to use precision medicine as a way to understand the disease better and identify treatments. And as you heard, I've been working on Huntington's disease for a very, very long time. So I've been working since 1989 at UCI on this disease. And it's absolutely devastating. It is... It just rips families apart and comprises probably some of the worst of all of these diseases. So it's been a real lifelong goal for me to try to figure out ways that we might be able to stop this disease. This was first identified in the 1800s by George Huntington. He was a medical student at the time. And he just gave a classic definition of the disease. He said it's confined to certain, unfortunately, a few families, so it's relatively rare. But not as rare as we used to think it was. It's about 1 in 10,000 individuals. Comes on gradually but surely, increasing by degree. So it's a progressive disease, just like all of these have progressive nature to them. And it's been transmitted to them in heirloom from generations way back in the dim past. So it's a genetic disease. It's inherited. And this is one of the key features about Huntington's is we know the mutation. So when we can study the disease in the context of that mutation, which we can't do for many of these other diseases. And in this case, one of the people who was probably the best known who had Huntington's disease was Guthrie, and he inherited that gene from his mother, and he had 8 children, at least 2 had HD. Arlo seems not to, he seems to have escaped it. 1 child died in a fire, 2 died, others died of Huntington's disease. So it's a classic example of this genetic inheritance in families. And it's characterized by symptoms of common chorea, only in an aggravated degree. So there are these dance-like movements, involuntary movements that are very characteristic of it, although the disease manifests in many different ways. Quite often individuals, we have many people in the community where one example is a woman went to her daughter's school and the principal's office brought her in and they basically said, you can't be here, you're walking around intoxicated on campus. Or if somebody will be arrested in the street, Guthrie was arrested for wandering aimlessly. And these are symptoms of Huntington's disease, but they're often accused of being drunk in public just because of these involuntary movements that they have. But it also affects, there's psychiatric symptoms, so it basically impacts emotion, thought, movement. Emotion, often depression. There's a very high suicide rate among people with Huntington's disease. Thought, so you lose some of your cognitive abilities. People can't take care of their families anymore. They can't perform their jobs. And this typically strikes individuals between the ages of, say, 30 and 50. So your prime of life, typically have young children. Your children are now 50% at risk. If you have that mutant gene, you just need 1 copy from your parent, you will get that disease if you live long enough. So you are in prime of life, and your children are now 50% at risk for the disease as well. And so it really is a genetically predetermined degeneration of nerve cells that are going to happen at some point in time. And as I mentioned before, this would be an affected brain and the significant loss. Now there's key structures that are lost called within the basal ganglia and general atrophy or shrinkage of the cortex, but it really affects the whole brain, which has made it quite difficult to treat and understand. And so what we'd really like to know is what is going on in those neurons that cause them to become so sick when they have that mutation present. So first you have to find the disease gene. So this is when I came into the Huntington's field, was before the gene was identified. I came in at UCI as a postdoc into John Wasmuth's lab, and they were part of a large international consortium to identify the gene that causes this. But this was really at the very beginning of the Human Genome Project when you couldn't even sequence a DNA, somebody's DNA. It was just starting to happen. And so there's 23 different chromosomes, about 30,000 genes. So it really was trying to find a needle in a haystack, trying to figure out where that mutation was in the DNA. But what there were families. So families and patients and all of this make a huge difference. There's a large population in Venezuela near Lake Maracaibo, and there just happened to be this founder that came. They often said it was Maria Concepcion that came down there, and because they would have large families, have them very young, it grew to be thousands of individuals in this pedigree who were either affected or at risk. So looking at these patient DNAs, it was identified actually the mutation in a 2-year-old little boy. So this disease can also strike children. And this is Nancy Wexler, who is my personal hero. And she really started this, her family started what's called the Hereditary Disease Foundation that brought together the scientists in a very collaborative form of research. And she went down there every year and would stay there for 3 months and work with these families, and they would do neurology exams. I would do pedigrees down there. And her mother had died of Huntington's disease, and her father was a psychiatrist to the stars in Hollywood, and he started this foundation. And so people like Frank Gehry, Julie Andrews, all of them were involved in this foundation. So that was, they helped find the gene, and more recently, really building on the fact that these people in South America have contributed so much, a number of HD families from South Colombia and Venezuela came and met the Pope. There was a day that he met with hundreds of individuals with Huntington's disease and their families and blessed them all. And the whole goal was that people shouldn't feel shame and stigma about the disease. As you can imagine, it was quite hidden for many years because of these implications. Your children are 50% at risk, you have problems speaking or look like you're drunk in public and other things. So these 3 individuals put this together, and it was a really pretty amazing thing that happened there. So back to the needle in the haystack, it was identified and found. And this was identified in 1993, reported in the journal Cell. And these are some of the individuals that were associated with this. And the authorship was quite unusual at the time, and even still, was the Huntington's Disease Collaborative Research Group. So there was no author. It was the whole consortium that was the author of this paper. And then each group was listed as well separately. But this was truly a collaboration where everybody brought their strengths, their knowledge together, and it's really influenced kind of how I see science and do science in my own life. This person right here is Francis Collins. He's the director of NIH, has been the director for a number of years now. And so he also has this passion for neurodegenerative diseases from all the work that he's had done on Huntington's, on cystic fibrosis, and other diseases. This was me a long time ago. So what is the mutation that causes Huntington's disease? This is a DNA helix shown here. This is what all our genetic information is, how it's encoded in this DNA molecule. And that then is transcribed to RNA, so you get a copy of it into an RNA, what's called an RNA molecule, and then ultimately protein is made. So these are the actual machinery of your cells that do things, but they have to be encoded by your genetic information. And so the Huntington's mutation is the very tip of one of the chromosomes, and it takes what's normally maybe 7 to 20 CAGs in a row, so this repeating triplet, this is a triplet repeat disease, this repeating triplet that grows past this point. When it gets 40 and above, it causes disease. Below 35, you don't get disease. So there's a mechanism in the cell that this has expanded. So it's a polyglutamine repeat disease because the protein part of this has a repeated glutamine amino acid. So those are the building blocks of proteins. So that's a Q. The symbol for that is a Q. So it's polyglutamine repeat disease. So basically you have this expansion of this region of the protein, and it changes the structure. So there's this very finite point at 40 where the protein just shifts and it changes. And that causes all these problems in a cell. As I mentioned, 40 and above causes disease. Above 60 causes juvenile onset. So young individuals get this disease. But there's still, even with that knowing the genetic cause, there's a lot of contributions from genetics, from the environment, that contribute to when someone will get the disease and how it will look in any given individual. So even something where we know the mutation, there's a lot going on that we don't know, and maybe influencing the fact that we haven't been able to treat Huntington's disease either. So, for instance, you might have someone that has 40 repeats and gets the disease at age 60, but there's individuals that it's a huge range of ages at the same repeat length. So there's a lot of variability that still is going on that we don't understand. And so there's been a lot of model systems. We can put the mutant gene into all sorts of different organisms. We can put it into yeast cells, into a worm, into a fly, into a mouse. And these have all taught us a lot. We've learned a lot from all of these, but we really need to look at the human context. Mice don't respond the same way to drugs that a human being does. They don't process it the same way. So we need to go into humans. How do we do that? We can't just go into humans. So the breakthrough really has been the production of these induced pluripotent stem cells. So this is when you have, say, a skin biopsy, for instance. So you take adult tissue. You can do this from blood, from skin samples, from hair samples, and they get reprogrammed back to what's called a pluripotent state. So these cells in a dish are almost reminiscent of an embryo, where they can just keep dividing in a culture dish, which you can't do if you take even a skin cell. You can't have it keep dividing. It's got a lifetime, but these don't. These just keep dividing. And you can differentiate them. You can have them go into becoming a neuron or a heart cell or whatever. You can guide them into that direction and use them to model disease, to use for drug screening, is something going to be toxic to the heart? Is something going to get across the blood-brain barrier? And so you really have an opportunity to develop new drugs to test these in these cells. This is just so this cell that's right at the top, or this little diagram. We're starting here with, say, with an embryo, just the equivalent of that. And with these cells, what normally will happen is you go from these cells into the skin, to the heart, to blood vessel. But when you reprogram those, you take those skin cells, you can reprogram those, they go back to that same pluripotent state, and now in a dish, you can make them go through development. So just these cells that came, so there's no embryos involved, it just comes from the skin, it gets reprogrammed back up, and now you can guide them to, almost like developmentally, into the types of cells you want to study. And you can do things like create a beating heart in a dish. So there's all sorts of opportunities now to use these to understand disease. A number of us were generating these iPS cells for Huntington's disease, and we, true to the HD community, started a large collaborative effort to generate lines and study them. And so we did that as there was groups all over the world, basically, that worked together on this and developed maybe 80 different Huntington's lines with different lengths of repeats, and have learned quite a bit from those. And so this really gets into this precision medicine approach. You have the patient, you can do all sorts of different big data analyses on those cells, and then hopefully learn something about the medicine that you need to develop and deliver back to the patient. So that's kind of the approach then that you can take using these iPS cells. And this is an example of something we recently did to study the blood-brain barrier in Huntington's disease. And so just to show you what that is.
The blood-brain barrier, such as other blood barriers, is a remarkable anatomical adaptation. Its most striking structural feature consists in endothelial cells, tightly joined by tight and gap junctions. These structures bind the cells side by side through several proteins, including connexins, claudin 5, and ZO1, thus maintaining the barrier's integrity. Pericytes cover about a third of the capillary surface, and surrounding glial cells, named astrocytes, support these capillaries with their foot processes. This capillary bed in the human brain has a total length of approximately 600 kilometers, with a surface area of about 20 square meters. It is permeable only to some molecules, such as nutrients entering the brain, or waste products, hormones, and excess neurotransmitters that leave the brain. Only a small number of drugs are able to translocate it.
So for Huntington's, we knew that something was going on there with the blood-brain barrier. It had been thought to be a little bit leaky, which is going to have huge ramifications, right? Things are going to get in that shouldn't, and other things not get in the way they should. And so we used, and it wasn't known whether this was a consequence of Huntington just being in these neurons, and this was a secondary damage, that things that were going wrong in the brain then communicated with the blood-brain barrier and made it damaged. But so we, this is from a very talented now postdoc in the lab, Ryan Lim, who used iPS cells to generate a blood-brain barrier. And in particular, those cells that they said that line the capillaries, the blood vessels there, these endothelial cells, you can differentiate into what we call induced brain microvascular endothelial cells, or BMECs, and model to see, is this barrier compromised itself in HD, and answer some of these questions that we had. And this is just an example of this barrier in a dish that came from patient iPS cells, and these proteins that they just mentioned that comprise the blood-brain barrier. And so we wanted to see what genes were expressed in these. And again, this DNA to RNA to protein, what are the genes that are transcribed and made into specific proteins in those cells, and is that disrupted? So using these omics approaches to try to identify what's going on in the cell, and these are just little bubbles of lots of genes that define various processes that's put together into a network. And what it suggested is there was a pathway, so that we really hadn't ever studied before, but it came out of this use of genomic data, a signaling pathway. So just it's a series of steps that happen in this particular cell that seem to be disrupted. And you could see even a difference if you looked at one of these proteins in a healthy brain versus an HD brain. You can see that very clear difference in the levels of those proteins. And what we found is that this seemed to compromise blood flow, that the production of blood vessels that contribute to what's called angiogenesis, or the creation of new blood vessels, and the flow, what keeps the blood flowing, was disrupted. And the basis of this was this set of genes. And so to model this in the dish, you could mimic that. So if you took a plate of cells, scratch a wound through them, they would start to grow back. And that was representative of this formation of new blood vessels. And the HD cells did this much more rapidly. So this is with increasing repeats. And they did this much more, they grew into that wound more rapidly than the unaffecteds. But because we had been studying that pathway from the genomic information, we took an inhibitor of that pathway, a drug, and found that that prevented that overactive formation of blood vessel mimic, and it restored it back to wild type levels. So studying that and using that kind of a precision medicine approach on patient cells gave us information we never would have been able to have otherwise. So it provided a model of the blood-brain barrier, but it has a lot of implications then. What drugs are going to be able to get across? Is that impaired blood vessel going to change how you deliver a drug? If you fix it, what happens? So there's a lot that we can learn from this and apply, and this now can apply to Alzheimer's, to Parkinson's, to many different diseases. So all of these disorders have quite a bit in common. They all have clumps of protein that accumulate in the brain, like the Lewy bodies. They don't get rid of the trash in the cells well enough. You have inflammation in the brain. So we then started working on looking at amyotrophic lateral sclerosis. And this is a wasting disease of skeletal muscle. It affects all the nerve cells in the brain and the spinal cord, from all these upper motor neurons all the way down, and then connecting out to the skeletal muscle. And it's not restricted to one body region. It spreads to affect many different areas of the body. And as these muscles deteriorate over time, and it's very short, unlike Huntington's, which lasts 10 to 20 years, ALS can last 30 months, average 30 months, 3 to 5 years. And they lose the ability to perform vital functions. They can't eat, they can't speak. Breathing becomes very difficult, and there are some cognitive effects in some people. And some of the sort of individuals this is also called Lou Gehrig's disease. And then, of course, Stephen Hawking's was, his life has been detailed in the movie The Theory of Everything. And the Ice Bucket Challenge for ALS to raise funds for ALS research has been incredibly effective and really helped fund a lot of the things that I'm going to talk about now. And just want to comment, this is Jay Fishman, who just passed away about a year ago. He's chairman of Travelers Insurance and had ALS. There's many different CEOs of various organizations, people that have had this really wonderful man who started this effort that I'm going to talk about, started raising money for it. And again, it affects all sorts of different processes. It affects the face and the tongue. You have more saliva produced, difficulty in communication. There's a general wasting, muscle weakness in the hands, ability to walk. It's hard to breathe, and very different emotions. It also affects psychiatric symptoms. ALS, and it's a little difficult to walk. You have these involuntary muscle movements. These are called fasciculations, so they have sort of involuntary muscle contractions that go on. And so unlike Huntington's, which is 100% genetic, almost all cases of ALS happen for unknown reasons. It's sporadic. It just comes out of nowhere in a family. Now some, maybe 15%, are caused by gene mutations, and as we, the scientific community, is learning more and more about what those are and using those to try to understand the disease. So these are just a few of the genes that you have mutations in that cause what's called familial ALS, which can be inherited, and the gene is known. There's also quite a bit of overlap that as these genes have been identified, where ALS has some overlap with dementia, frontotemporal dementia. So there's some cases of ALS that are pure ALS, pure motor neuron, and some cases of dementia that are pure dementia, and then there's this spectrum in between. And so trying to understand that and treat that is very, very complex. But because there are these mutations that we can generate IPS from cells from patients, we can start to learn more about this. And just to note, these are all the drugs that have been tried in clinical trials for ALS, and none of them have worked. There are two that give just a little bit of an effect, but not much. And this is also, again, because we don't understand the disease well enough, because there's so many different subtypes of individuals who have ALS. And so to start to get at this, we were funded by NIH to form a group called Neurolinks. It's one of 6 link centers around the country, some studying cancer, and we're studying motor neuron disease. And it's a group of investigators, again, this whole collaborative research effort where you leverage what people know how to do and work together. And what we're trying to do is understand what is going on in an ALS motor neuron from a patient. What are all the things that are going on so we can identify a signature that then we can see if we can get back to normal. And this just shows the sort of pipeline of this group. This is just a small representation of the people involved, where we have these patient-derived cells, turn them into motor neurons, which are the affected subtype in the disease. You could do this with a dopaminergic neuron for Parkinson's. You could do this with a hippocampal neuron for Alzheimer's. Same general principle would apply. Do all these different assays. So this is looking at RNA, all the RNAs, all the proteins, what the DNA structure looks like, what the cells look like over time as their neurites start, those processes I showed you, they start to retract, the cells start to die, you can follow those over time, and then put all that together in some way to get something meaningful out. And so this is just an example of the motor neurons, what they look like in culture. And so we distributed, there's a core at Cedars-Sinai that grows them, differentiates them, then gives parallel cultures, so you're looking at the same thing to all the different labs. And some of the challenges, so we find differences. So you find differences in these cells. In this case, it's the C9 mutation, a particular known, fairly prevalent mutation. And you find, if you just look at those colors, you can see a bunch of blue and then red and then blue. So one of these is ALS, a set, and one of these is controls, is unaffected. And you see this for all of these different assays. But it's very difficult to go this way to try to figure out what overlaps. It just doesn't work. So what you end up with is just a hairball of information, and you have to try to make sense of that. And so for this, there's a bit of magic involved by computational biologists that I don't even understand, that called omics integrators. So putting all this information together and getting something meaningful out, which doesn't look all that meaningful. There's all these little bubbles of networks that you can then study. But now we have things to go after. We have things we wouldn't have known to even try to go after before. And so we're starting to tweak those systems and look at them in human brain, look at them in fly models and other models, and see if they make a difference. So building on this, though, and this took years of just putting the pieces together for this endeavor, trying to figure out what to do with all this data. So there's a program now called Answer ALS, which is a precision medicine approach. And it's really to look at an individual's genes, their neurons, and their brain to try to identify individual ALS treatments. So that's Team Gleason started this effort, and Steve Gleason is an NFL player who has ALS, and they started some of the fundraising. Then we started working with Jay Fishman from Travelers, and somebody from American Airlines, and somebody from here, and so this has really taken a village. And there's a number of individuals and organizations where they have high rates of ALS associated with them, and they've really partnered in this effort to use precision medicine approaches. And so the general overview is now way beyond what I was talking about with Neurolinks, although that is at the core. So this is all the various genomics that I was talking about using iPS cells. But in this case, we're taking 1,000 individuals with ALS, both with known mutations and sporadic, making iPS cells from each one of those, so getting blood samples, generating iPS cells, taking that through that whole pipeline, as well as getting DNA sequenced from the New York Genome Center, all sorts of clinical collection, imaging, wearables, iPhones, smartphones that they can tap in, they can monitor their symptoms through an iPhone. Biobanking of tissues and blood, and then this large analytics effort, so using computers to try to understand patterns that might be showing up in subtypes. So far there's over 1,100 patients, over 100 investigators, so that, again, this is this large collaborative effort that UCI is one of the sites for, and I should say the transcriptomics, the RNA, that's being done by the Genome Center on campus. And so the idea is to get the right medicine for the right patient. And this can be applied to all these diseases, FTD, Alzheimer's, Parkinson's. And there are efforts. They have accelerating medicine partnerships for Alzheimer's, accelerating medicine partnerships for Parkinson's disease. And it's a massive collection of samples, CSF, plasma, blood, and so far there's over 27,000 samples that have been gathered to do this. And matching then ultimately autopsy material and spinal cord tissue with the biology we learn in those cells from those same patients. So this really is getting at what happens in the individual. And then, as I mentioned, trying to see what holds up, what's true, what's valid. And so we hope to get personalized therapies for ALS. And as I mentioned, this applies to everything. So Alzheimer's disease is a hugely prevalent disorder and affects millions of people across the country. And again, there's many things that affect it beyond genetics. So genetics is very critical. There's susceptibility genes. There's again the same thing. Most of the cases are sporadic with no known cause, but there's many things that increase your risk and things that decrease your risk that we want to understand how do they contribute to the disease in an individual. And at UCI, there's this amazing institute called UCI Mind. It has a number of investigators, and they have an Alzheimer's disease research center. And so they've amassed hundreds of tissues, biosamples. They have an IPS core to generate IPS cells from individuals with Alzheimer's disease, and many, many researchers and clinicians who are poised to do this type of precision medicine approach and have this major interaction with UCI Health and using these types of approaches. So we're really excited about working on these types of problems all together as a community, and with you all, with the community here in Orange County, so that we can really find treatments for these diseases. This just, again, they have cores for tissues, for data management, to generate IPS cells, for outreach to the community, and there's an Alzheimer's clinic. So we hope at UCI then to use these kinds of approaches to really find individualized treatments, to use precision medicine and develop precision medicine with the community, and get all this information and use machine learning, so sort of artificial intelligence, machine learning, to put that into something that allows us to understand what causes the disease, what are the things that influence the course of the disease, and allows us to ask, what if? What if we do this, will it impact the outcome? And that's really where we're going with all of this, and I just want to thank you very much for your time and attention. Thank you. Yes?
Just a couple of quick
Yeah.
questions. Do you think Hawking does not have ALS in that he's survived for
Yeah,
decades, if not years, well past the 30 months,
Right.
not showing signs of dementia, so maybe he doesn't have ALS?
He definitely doesn't have classic ALS, but he has some subtype. I mean, a lot of the clinicians have commented on that he has some variant of ALS, and it's an average of 3 to 5 years. You still see people who go 10 years. You see people who go 1 year. It's usually rapid. Oh yes, thank you. Yeah, whether Stephen Hawking's has classic ALS. Yeah. Did you have another question? Did you say?
Well, a friend who unfortunately is suffering much more quickly.
Yeah.
It seems to be fairly advanced in just a few years in. And he was in the military, and he feels an incredible amount of shots that he had to take in the military for some reason. I don't know. He seems to think there's some connection to all these needles and shots that he was exposed to.
Yeah. So the question being, were there things that could have affected your friend, like shots or needles, and there's clearly environmental, which encompasses a lot, pesticides, your lifestyle, all sorts of things, but that could It's entirely possible. And that's what the kind of thing so you want to track that history, right? So let's say you got an IPS cell, you made IPS cells from that person, you know this history, you know all their clinical symptoms, and you try to understand what connections there might be. Yes?
Do you see commonalities? What kind of environmental structures? Do you see commonalities in patients with ALS or Huntington's chorea that have similar environmental causes?
So are there similar environmental causes across these various diseases? So that's really just beginning. So you know that stress affects Alzheimer's, stress affects Huntington's disease, stress affects ALS, exercise benefits all of those. So we're beginning to learn those things, and they're clear, and inflammation, if somebody has an illness, sometimes that'll help precipitate Alzheimer's disease, for instance. Like my mom has Alzheimer's and she was very ill, and then that's when it started. And so so there are clearly overlapping things, but there will probably be, and pesticides, that's another one that's come up for both ALS and Parkinson's disease as potentially contributing, but it hasn't been really proven necessarily. So there's that research. This I think is a way we might be able to get at that. Yes?
Do you see any, I'm a psychiatric nurse, so I work,
Oh, okay.
I work with UCI.
Oh, okay.
Psychiatry. Do you see any of the similar diseases like illness like schizophrenia or bipolar?
So the question, do we see, what with schizophrenia and bipolar?
Is this coming up perhaps under this umbrella, or is this something?
No, so absolutely, and I didn't put this here, but these same approaches can be used for schizophrenia and bipolar, and there are clearly some overlapping pathways as people start to look at the genomic information. Very distinct ones also, but those are the kinds of things we want to learn. Yes?
Thank you very much.
Oh, thank you.
Fascinating.
Thank you.
I was wondering, I know UCI does a lot of research in a lot of different areas.
Mm-hmm.
And I was wondering about how you work together with other research departments. For instance, with other genetically based diseases, such as type 1 diabetes.
Yes.
You know, you're doing research.
Yes.
And the question of the role of pluripotent stem cells.
Yes.
So I'm wondering if you could say anything about
Yeah.
you work with other diseases.
So the question being, how do we work together at UCI on various diseases that might seem extremely distinct, I guess, right? But so for one thing is at the Sue and Bill Gross Stem Cell Center, we have investigators working on blood diseases, on diabetes, on neurological disorders, on all sorts of things, and we regularly interact because there's still so many, even though we're differentiating to a neuron, or we're differentiating to a pancreatic islet cell, or we're differentiating to there's so many things that are common about the approaches, so we really try to learn from each other. And these centers and institutes, so UCI Mind is made out of people in psychiatry and neurobiology and behavior, from neurology, from all sorts of different disciplines. They work on Down syndrome as well as Alzheimer's. And so all of these work together. But one of the things actually Suzanne and I are working on with this precision health is to bring together, or at least to figure out ways and strategies that we can work together very closely with, so the cancer researchers can work closely with the people doing neurodegenerative disease, that the groups doing all of us and identifying the genetics that's going on, so that you have that, because it's really powerful when you can do that. Makes a difference. Yes?
Now that you have the endowment from Samueli and the Integrative Medicine, have you guys started doing any type of research with the mind-body movement and prolonging the quality of life for any of your
So I don't know the answer to, so this is whether with the Samueli gift, if this integrative health is moving forward. And it's really being planned, and I know they've already had the Samueli, a center that started doing these types of putting all of those pieces together. I'm a PhD in the lab, working with patients, but they are moving forward with these things.
I'm thinking, have you seen any benefits with the mind-body movement helping prolong the quality of life for people who have been diagnosed with these?
Yeah, so have I seen benefit from those kinds of a mind-body movement type thing? For sure, there's now formal studies on, say, exercise and its effect on Alzheimer's, or cholesterol intake and, nutrition, right? And then does Tai Chi help? All those things are being formally studied now in clinical trials. Have I seen it? Yeah, I mean, there's lots of anecdotal things, and I think it's really important personally, but there hasn't been the evidence that I've seen to support that, but I have anecdotally seen where things like that help. But yeah, I don't know if you have any comments on that, Suzanne, or yeah, okay. Yes. It is a form of dementia. So dementia is kind of the broad net, and then there's Alzheimer's disease, which is a form of dementia. There's frontal temporal dementia, which is a form of dementia. There's Lewy body dementia. There's dementia with Huntington's disease. So they're subtypes of dementia, okay? But they overlap. So you can find alpha-synuclein, for instance, that form Lewy bodies in Alzheimer's disease and in Huntington's disease. So those things can accumulate, but they're sort of distinct. But that's kind of one of the problems, is where does that line fall, and what commonalities are there, and what differences are there? Yes. Yes.
I'm curious about the process to make a skin
Sure.
a pluripotent cell.
Sure.
So I mean, you can't do one by one. There must be some machines. It must be really tiny. What's the process?
Yes. So, what's the process to generate iPS cells? For instance, if you take, we're now using blood, but let's say classically you would take a little punch biopsy. I had this done, a little skin sample. You'd grow those cells in a dish, okay, so that you had a nice big full dish of those things, and then you'd add these factors. So it's like a little cocktail of factors that reprogram. So sometimes they deliver them with viruses. We're trying, other way you can do them with drugs. You can do it with a number of different approaches to turn those back the clock, basically turn them back into iPS cells. But you're right. It's quite a process. It's not an easy process. It takes a couple months, and what you find, you look for those cells that got those factors in and keep proliferating, and then you select out individual groups of those cells, and those become clonal lines. Then you expand those in another culture dish until you have a cell line that you freeze back in vials, and then you take them out when you need them. And so it starts from a single colony, single clone in a dish that you then expand and grow. So it takes months for each one of those. Yeah. So there's companies that are trying to figure out ways to automate that, and I think it will become automated as we go along. And Cedars certainly has a process that's very efficient. Yeah. Yes.
Yeah, I'm wondering, you said earlier that some of the clients that commit suicide, is there some way you have a routine approach to somebody's psychiatric status to sort of, identify those people and work with them?
So that's a great question. So is there a way to identify those who are at higher risk for suicide and prevent it, right? Right now, they are seen by, well, it's interesting. Sometimes the first person somebody with Huntington's disease will see is a psychiatrist because they have major depression. It's part of the disease. And they will present that way, or you often hear families say, Dad's personality changed, or Mom started yelling all the time. Just things would change. So you'll see those symptoms, and they'll get treated for depression, so that they'll get antidepressants, and that can help the depression. But you can't really predict necessarily who's going to choose to commit suicide. And that's one of the things we'd like to find out. Yes?
And also, I'm curious about, how and where along the way are their emotions sort of stunted?
Yeah.
So that
Yeah, unfortunately. So so what you're saying is kind of as you go along in the disease, do you reach a steady state, almost a place where you aren't as depressed anymore as you were early on as things start changing? And so classically with Huntington's disease, what you see, first of all, the disease we know starts years before you start seeing the symptoms that would cause a diagnosis of Huntington's disease. So things start changing over time. People, the psychiatric symptoms or the cognition starts to change. But it doesn't go away. It will continue, and it's horrible. But I can't tell you the number of cases that I'm very familiar with where, they just don't have the same perception of what, what's going on. So Huntington's very different than Alzheimer's. Alzheimer's, you have no memories left. You can't recognize your family. You don't know what's happening to you. Huntington's, you're very aware of what's happening to you. But there's things going on in your brain that you cannot control. We know somebody set fire to themselves and killed themselves that way. Somebody that was well advanced in Huntington's. And it can happen in any stage of the disease, really. There's a point when they no longer physically can, and they're bedridden, right? So then they can't. But beyond that, it's kind of a continuum of those symptoms. It's a pretty awful disease. So, yeah.
I have a friend that was diagnosed with cortical Degeneration.
Yeah.
And she was assisted living, and within a 2-year period, she's bedridden.
Yep.
And basically paralyzed up one side.
Yep. That's sometimes that cortical, what's it called? Cortical basal degeneration. Sometimes diagnosed as Huntington's disease or Parkinson's disease.
Initially, they thought she had Parkinson's.
Yeah, that's often.
And they checked the test.
Yeah, and it wasn't Parkinson's disease. It's awful, and it's very similar to. It looks a lot like Huntington's, aspects of Huntington's and aspects of Parkinson's disease. Yeah, it's a really, and it's again, it's mostly sporadic. There's not a known genetic cause, and it's not understood what's happening. So those are also people those patients UCI, trying to get blood samples from, or tissue samples, create iPS cells ultimately. So that would be a goal for those kinds of things, is to use these approaches to understand even very rare diseases like that. Yeah.
Realistically, do you think breakthroughs are decades away, or realistically?
No, again, I've been working on this disease since 1989, and so when the gene was cloned in 93, we all said, got the gene now, we're going to be able to have a treatment in 5 years, which obviously hasn't happened. But I personally feel that we are truly, and I'm saying this absolutely sincerely, at a time of unprecedented hope, to be able to have these ways to study it that we've never been able to do. I felt like this, the last time I felt like this was when the Human Genome Project was in place and you could sequence DNA and you could find a mutation. I feel like we're at that stage again of where the technology is just going to explode, and we're really going to find things that are going to make a difference. In the next, it takes 10 years, 5 to 10 years to get a drug to the market, right? There's a lot. There's phase 1 safety trials, there's phase 2, there's phase 3. But one of the things, say, Dr. Fedorov is very motivated to do is use repurposed drugs. So things that have already been through safety trials, that have already been used in patients that you could then use. It was an indication X, but now you can use an indication Y because you found out that this subtype of patients will likely respond to it. So that's going to speed things up also. So I do feel like there's a lot of hope now. Yeah.
I just have one question.
Yeah.
Do you know if support for NIH is continuing, because it's a precarious environment out there?
Now there's a question. But yeah, how's NIH support look? Didn't look as good yesterday, right? But No, it's, there's been a brain initiative that's been funded that really has stimulated research funding for things like Alzheimer's. And so that's made a big difference. There's been funding for that. It's always difficult, and it's really difficult to get funding for some of these more rare diseases. And they're still very underfunded compared to cancer research, for instance, which I'm not, that's important also, but very important. But, it is an issue. So things like this Team Gleason, where people come together, and philanthropy is huge in trying to do research for these diseases as well. Yeah. Yes.
Do you think they're going to meet the 2025 initiative on Alzheimer's?
Will they reach the 2025 initiative on Alzheimer's? I don't know. I hope so. It's hard to predict sometimes. I think that we're making significant, making significant progress. But I always hesitate with that because for so many years we've had patients ask us, so when are you going to give us a treatment? And so I'm hopeful though. Okay, thanks. Yeah.
Dr. Thompson.
Thank you.